Okay, the first item in chapter 4 is about magnification and microscopes. So magnification is the ratio of the image size over the actual size. So when you use a lens to observe something, it's increasing the size of the image. So let's say you're looking at an ant with a 10x lens, and the light that bounces off the ant goes through the lens and it gets refracted, meaning the light is bent. And it bends in such a way that the image is larger. So if you were looking through the 10x lens, let's say you took a picture of the image and you measured that image, it would be 10 times bigger than the actual size of the ant. So that's why we call it a 10x lens. And there's 3 types of microscope. So the first type is the light microscope. And the light microscope, however, is limited because it uses visible light to form the image. But the human eye can only see light that is larger than, has a wavelength greater than about 380 nanometers, or 380 billionths of a meter. So any light that's has a shorter wavelength than that is not going to be visible with the human eye, and so this is a limitation. And also there's objects that are smaller than 380 nanometers. For example, viruses are smaller than that. They're about 100 nanometers. So you can't see a virus with a light microscope because the light is simply, the wavelength of light is too long. But you can see viruses and other small objects inside cells using an electron microscope. So the electron microscope uses electrons instead of visible light. And the wavelength of the electron is much shorter. The electron's wavelength is only about 1 nanometer. And so it can resolve much smaller objects. So you can form images of viruses and other subcellular structures using the electron microscope. And there's 2 kinds of electron microscope. There's the transmission electron microscope, and that sends the electrons through the specimen, through a very thin section of the specimen, and then that forms an image, and it's good for seeing small details. And the other kind is called the scanning electron microscope, and in that case the specimen is coated with gold or some other metal that reflects the electrons off the surface, and so it's good for observing surfaces. Another important concept in chapter 4 is the cell theory. So this theory states that all living organisms are composed of cells, and also that cells are only derived from existing cells. So there's no spontaneous generation of cells from non-living matter. It's always cells being derived from existing cells. And another concept is cell size. So cell size is limited by the surface to volume ratio. So why are cells microscopic? Why aren't there any giant cells as in science fiction movies? Well, because as the cell grows, its surface to volume ratio goes down, and that limits the transport of nutrients and gases. So the cell has to bring in nutrients, and it has to exchange gases, has to bring in oxygen and take out carbon dioxide, and that's a passive process, and that process can only go so fast, and so as the surface area of the cell decreases, that becomes more and more difficult to do. There are two types of cells. There's the prokaryotic cells. These are cells that have no nucleus. And the eukaryotic cells, these are cells that have a nucleus. So there's of the prokaryotic cells, there's bacteria and archaea. And for the eukaryotes, there's 4 types of eukaryotes: the animals, plants, fungi, and protists. Membranes are composed of phospholipids. So the membrane has these phospholipids, and the phospholipid is similar to the triglyceride, except that in the case of the phospholipid, there's only 2 fatty acid chains, and the 3rd carbon of the glycerol has a phosphate group and a choline group attached to it. So here we have the 2 fatty acid chains, and the hydrocarbon part of that is hydrophobic. So it avoids water, whereas the phosphate group in the choline is hydrophilic, and so it will associate with water. It'll interact with water. So we can represent the phospholipid as having this polar part, we call it the head group, and then the 2 hydrocarbon tails, the hydrophobic part. And if you have phospholipids in a layer like this, if you have 2 of these layers, it forms what's called a phospholipid bilayer, and that forms the membrane of the cell. So outside of the cell you have an aqueous exterior, and inside you also have an aqueous interior of the cell, and the hydrophilic parts of these phospholipids point towards those aqueous compartments, and the hydrophobic tails face inwards. Plant and animal cells differ in a few traits. One is that the plant cell has a cell wall, whereas animal cells do not have a cell wall. Plant cell also has a vacuole that stores water, and it also has chloroplast. Bacteria, archaea, and fungi also have a cell wall. And there's 2 organelles, the chloroplast and mitochondria, that are different from other organelles in that they're believed to have been originating as free living bacteria that were engulfed by a larger cell to become endosymbionts. So they developed a symbiotic relationship. The chloroplast is the organelle that does photosynthesis. The mitochondrion is an organelle that does cellular respiration. So we'll see this in more detail later. Basically, cellular respiration is how cells get energy, acquire energy from food. And the evidence for the endosymbiont theory is, first of all, that the chloroplast and mitochondrion have their own DNA, unlike other organelles. And also, the mitochondria and chloroplasts have double membranes. So the idea is that the inner membrane came from the original bacterium, and the outer membrane is from the cell that engulfed them. Another line of evidence for the endosymbiont theory is that the mitochondria and chloroplasts more closely, their DNA more closely resembles that of bacteria compared to eukaryote DNA. And also the mitochondria and chloroplasts have their own ribosomes. And those ribosomes resemble more closely the ribosomes of bacteria. Also, in chapter 4 is the cytoskeleton. The cytoskeleton is composed of protein filaments, and there's 3 types of filaments: the microfilaments, microtubules, and intermediate filaments. And the microfilaments are composed of a protein called actin. Microtubules are composed of tubulin, and the intermediate filaments have a number of different proteins, and they tend to be very fibrous proteins and also proteins that coil up and form a kind of coiled coil structure. And the microfilaments are involved in cell movement, and you find those in the cytoskeleton. Microtubules form tracks that organelles can move along in cells, and they're also involved in the formation of the cilia and flagella. And intermediate filaments have more of a structural role where they support and anchor the organelles and maintain the cell shape. Finally, in chapter 4, we have a summary of the organelles and their functions. And the organelles can be categorized into 4 groups based on their function. So the first group is genetic control, and these are the nucleus and the ribosome. Nucleus houses the nucleic acids, the DNA, and the RNA, which control the cell, and the ribosome that synthesizes protein. Then you have manufacture, distribution, and breakdown. So that's the endoplasmic reticulum, which modifies proteins for export. The Golgi apparatus, and that also is involved in the export of proteins, as well as distributing proteins within the cell. The lysosome and the peroxisome. In the energy processing, so there's the mitochondria and chloroplast. These are involved in acquiring energy for the cell. And then finally, the structural movement and communication. So you have the cytoskeleton, those filaments. You have the cilia and the flagella, which are involved in movements of the cell. And communication, that's the extracellular matrix, as well as the cell junctions.